Today we have a very special guest, Dr. Adalja. He's a faculty member in infectious disease, emergency medicine, and critical care. This is pretty impressive. And at the UPMC Center for Health Security. And we'll hear about the Ebola epidemic. So, thank you.
Thanks for having me. I'm glad to kick off this series with this very timely topic. I talk very fast, so raise your hand if I said something that you don't understand, and I'm happy to take questions about this and any other weird emerging infectious disease that you might have. So the title of my talk is this Ebola outbreak in West Africa? Is this a familiar pattern, or is this something new about Ebola? And that's been a big question that's been in the media a lot. And I'm going to try to address what we know about Ebola. So when people think of Ebola, most people think of this movie, and I think it's a great movie. I've watched it so many times that I can't count. It's outbreak, and it really got a lot of people interested in infectious disease. And there they have a virus there called the Microbiology, which is a surrogate for Ebola. And some of the major differences there, and this is what conditions how people think about Ebola is not airborne. And it does not liquefy organs like Cuba Gooding Jr.'s character says in this very dramatic scenes that it liquefies your organs. And that's what most people think, that it does liquefy your organs, and it's this dreadful disease, but it is a very explainable, non-magical type of disease. And not every case is hemorrhagic. Not everybody bleeds out. Only about 50% of people have hemorrhagic fever with Ebola. That's why they changed the name now to Ebola viral disease from Ebola hemorrhagic fever. But there are definitely some similarities that you see with Ebola and in the virus portrayed in this movie. There definitely is a zoonotic component. By zoonotic, I mean it comes from an animal and goes into humans. And that happens very dramatically in that movie. And that's what happens with Ebola. And I'll talk a little bit about what some of those animal species may be that transmit this virus to humans. There's definitely a role of travel. In that movie, you see people traveling all around the world, and that's an important part of emerging infectious diseases, that they can move with people from one side of the globe to the other. We've seen it with MERS, we've seen it with H5N1, we've seen it with multiple different viruses, so it's important that even here in Pittsburgh, or wherever you may be, that you're thinking about what viruses are occurring on the other side of the planet and being prepared to look for them. And there also were calls for quarantine in this movie. This movie is very dramatic. They want to bomb a whole city. That's not something that's happening here, but we did see a massive quarantine go on in the West Point area of Liberia, where people were shot for running out of that quarantine. So that happens with every type of epidemic. You can even remember during H1N1 when there were calls for President Obama to shut the southern border. So that's something that happens with anything that's unknown, and I think that outbreak does serve some purpose in priming you to think about Ebola. The other movie is people think about this, is World War Z or the zombie attacks. Ebola doesn't cause zombies, but there was a lot of interest in this because the CDC had a program, a preparedness program, to get people ready for a zombie attack, because if you're ready for zombies, you're ready for everything. But it's been backfired a little bit, and I did some press calls, and I actually did a radio show on KDA here, and I was asked about what happens if a plane full of people with Ebola gets shot out of the sky and a body part lands on you, which is actually like a direct scene from World War Z. What happens? And I think that's the least of your worries. If body parts are falling off of the sky onto your head, I think Ebola is the least likely thing to worry about, rather than, traumatic brain injury would probably do you in. So this is what I'm going to talk about. I'm going to first start out with a snapshot of West Africa and what Ebola is like and what is going on in West Africa. Then I'm going to give you a little bit of the history of Ebola and other filoviruses, the family that Ebola belongs to. I'm going to talk about the microbiology, the epidemiology, and the viral ecology, how this virus interacts with people and other species in the environment. Talk about some of the clinical details of cases of Ebola, how this infection causes such severe disease. Talk about some of the treatments that you might be hearing about in the media a lot, especially with ZMapp, for example. And then I'm going to go back and revisit the West African or world situation to end. So Ebola in the West African situation. So this began really in December of 2013, and this is the Zaire strain of Ebola. That's what I mean by ZEBOV, if you see that abbreviation. And what's unique about this is West Africa really didn't have an Ebola outbreak in the past, and that this outbreak occurred in the epicenter. The epicenter of the outbreak is really on the convergence of three border areas. Anytime you have a disease that crosses borders, which a lot of them do, it becomes very difficult, especially in an area where you have three different ministries of health, three different governments, a whole host of communication problems that occur when you're dealing with three different countries trying to stop the spread of a virus that is easily traveling amongst people that move across this border freely. So it's Guinea, Liberia, and Sierra Leone. We've seen secondary spread to Nigeria with the one traveler, and also in Senegal after somebody escaped quarantine from Nigeria and went to Senegal and was diagnosed there. This outbreak has seen medical evacuations. We know of 3 evacuations to the U.S., 1 active patient right now in Nebraska, 1 to Spain where a priest died, 1 to the United Kingdom, and one to Senegal. There was one indigenous case to Senegal that moved from Nigeria to Senegal, but one case of evacuation to Senegal. So far, the last time I checked the numbers, we've got about 3,685 cases with 1,841 deaths. So this is about a 50% or so mortality. This is a little bit less than what we've seen in other Ebola outbreaks. It's usually, with the Zaire strain, it can reach up to 90%. So this is a little less severe. This may not be a true number because there is a lot of issues with case finding there, and I'll talk about that later on in the talk. So now moving to the history of Ebola. This is the name Ebola comes from the Ebola River, and if you see here where this is a map of the DRC, and that's the river where the first outbreak occurred. And that outbreak occurred in 1976, almost simultaneously with another outbreak in Sudan with another strain of Ebola called Ebola Sudan. So these both came onto the scene in 1976. But we had known about this family of viruses and knew how dangerous they were before, because in 1967 there was an outbreak of Marburg virus, and that was Marburg comes from Marburg, Germany, where some African green monkeys were imported to Europe, and there was an outbreak there. 7 people died, so we knew that filoviruses had a high capacity to kill. And then Ebola came on in 1976, was the new filovirus that had been seen. And it's important to remember that so we've had these viruses for a while. People have been working on them for a long time. There's this impression now that all this is being accelerated, but this is really stuff that's been going on for decades. In 1989, a very famous episode happened in Reston, Virginia, where a monkey shipment from the Philippines was found to be infected with Ebola Reston. Before this, every filovirus had been deadly in humans. However, the Reston virus was not that, it infected probably about 6 humans there. Nobody got sick, they just seroconverted their antibodies. I actually last week talked to one of the physicians who took care of one of the patients in Fairfax, and it would have been it was a very scary time, but luckily this virus isn't pathogenic for humans. In 1994, the Côte d'Ivoire strain, or now it's called the Thai forest strain, was found. This was just an individual who had been around a dead chimpanzee and got infected. That's important, the dead chimpanzee thing, I'll get back to it. So that was another strain that was found. And in 2008, the latest strain was found. It's called Banda Bagyo. It sounds kind of odd, but that's one of the other strains that's occurred. So there's a whole family of Ebola viruses. Zaire is the most deadly, and that's what we're dealing with now in West Africa. This is a slide just showing you where Ebola occurs. And what I said earlier was that West Africa never really had that many outbreaks. Notice this is 2012, so this is before this outbreak. Ebola usually occurred in countries like the DRC, Gabon, Uganda, and in Sudan. And they're very adept at stopping these outbreaks. That's why when this outbreak occurred, we all assumed that this would be extinguished, because every single time this has happened, simple measures have stopped it. And it may be because these countries were so adept at doing it, because they've been doing it since the 1970s. So moving to the microbiology of the virus. Phytovirus comes from the word thread, or filament. So it kind of looks like a filament here. When I was younger, I was obsessed with this virus. I had this on my parents' fridge. I hung this picture up because I thought it was such a cool virus. So there's 2 members, Marburg and Ebola, like I talked about. There are 5 species of Ebola, which we alluded to. It's a negative sense RNA. That means that you have to make another copy of it to make copies. And just a general, it's probably not that important to everybody, but I think anything that's negative sense is cool in general. Those are the most deadly viruses that tend to be negative sense, and the DNA viruses in the positive sense are not as interesting to me. It's envelope. That means that if you leave it outside, it dries up, just like because it's made of like this lipid fatty layer, this envelope, and it can dry up. So it's not that hard to clean up. If you do have to clean it up, you don't have to do the same thing you have to do for C. diff, for example, which forms spores. It has 7 genes in it. I'll talk a little bit about these genes because these genes are important for how this disease causes illness. So this is a list of the genes. So the nuclear protein, there's the virion protein 35, which is an interferon antagonist. So those of you know that interferon is a major part of the host defense. The Ebola virus is very adept at subverting that, and that may be why it can cause such severe disease. I want to draw your attention to the glycoprotein. That's how it binds to receptors and causes membrane fusion. Interestingly, Ebola has a soluble glycoprotein as well. It makes one that sits on the spike, and then it also can secrete one, almost like a toxin. And what does it do? It binds up the antibody that would be binding the virus and causes immune activation. So this virus is very adept at causing disease in humans when it gets into other things are RNA polymerase. That's a target for new drugs. Most of our other stuff are directed at the glycoprotein. It's just a picture, another picture showing you how these all fit, and this is the glycoprotein spikes, and then all of the other things that are associated with the nucleic acid. So moving to the 5 species, just in summary, there are 5, like I said. 90% mortality is what we've seen with Zaire in the past, about 50% currently. Sudan, 50%. What's going on right now in the DRC, they have another simultaneous Ebola outbreak. It's a hybrid strain between Zaire and Sudan, which is a simultaneous unrelated outbreak. Reston, 0 mortality, 4 seroconversions. I said 6 are actually 4 that seroconverted. Bundibugyo, 40% mortality. In Cote d'Ivoire, we had 1 case, so we can't really talk about how the mortality goes with that virus. Most diseases, though, are really caused of the phytoviruses are Marburg, Zaire, and Sudan. Bundibugyo and the Cote d'Ivoire strain really haven't returned, and we know that Reston is not pathogenic in humans. So when we talk about phytovirus infections, we're talking about these 3 viruses specifically. This is a good slide kind of showing you where this virus has been, and I want to illustrate something. So this is Reston. Reston has been imported multiple times in the United States. You see in Virginia, Texas. We had an imported case of Marburg in Colorado, which is very important when people say we've never had these viruses in the United States. First of all, we've had these viruses for a long time because we've been working in labs with them. But we've had imported cases of Reston and Marburg, which is as close as you can to having a live case of Ebola in the United States. And remember, this was not even recognized until after the fact, and the woman said, I think I might have Marburg, and they tested her antibody. So she was unidentified, and she was taken care of in a hospital, and there was 0 secondary transmission. And we've also seen, in Europe, it's been much more common. Marburg, obviously, has been there multiple times. Just another slide kind of showing you exactly where these are all the Reston importations in the United States that have occurred, usually tied to infected monkeys. This slide might not project well, but this is kind of a tally of all the Ebola outbreaks we've had in the past. The largest one before this one was the one in Sudan, where we had 425 cases with a 53% fatality. Again, it was a Sudan strain. So this one really has broken all the records. But one other thing I want to draw your attention to is if you add all of these up, it's still not that much, even adding on what we have now. Ebola is still a small public health problem compared to malaria, tuberculosis, HIV, influenza. And that's a lot of the reasons, that's some of the pushback we're getting on why aren't there antivirals. The fact is that this is not a huge public health problem, and it has never really been. And this outbreak, even though it's the biggest, still doesn't make a dent in the number we see from people that are dying of malaria right now. So what is Ebola, and how does it occur in humans? So I think of it as a spillover disease. It's a zoonosis from an animal that spills into humans. We don't really understand exactly how that happens. Humans are really end hosts because the virus does not transmit from human to human very efficiently. It's not like measles, not like tuberculosis, not like flu. It's not well adapted. We find antibodies in bats, and they may be a reservoir host. There may be others as well, because it's unclear when bats were infected and how long they've had this virus. But bats are our chief suspect, but there may be other reservoirs that exist. And the reason why people think about bats as reservoirs for viruses is that they're everywhere. It's one of the most populous mammalian species. They can fly, they can go everywhere. And we know that other viruses that are deadly, like SARS, probably MERS, rabies, the Nipah virus, the Hendra virus, all have bats as hosts. So it's not uncommon to look for bats to find new viruses. But we've seen no documentation of transmission directly from bats to humans. So that's why we still have some ifs about what the reservoir is and how humans get infected. So we also don't know why these Ebola outbreaks come and go. They seem to be explosive. And what we think happens is that there might be some activation in the reservoir host. The hosts get pregnant? Are they stressed? Is there a change in their food source that causes the virus to replicate higher and then spill into human species? And that's really still a hypothesis that needs to be worked out. So what's some of the hypothesis about the spillover? What is the trigger? These outbreaks are rare. Is it because we're contacting the reservoir very rarely, or is it a rare animal that has it? Again, bats are ubiquitous, so this kind of fits with the whole idea that bats may not be the ultimate reservoir. There's an interesting way that people think about Ebola outbreaks, and it kind of goes to the wave-particle duality that people talk about in physics, for the one physicist in the crowd that I know is sitting here. So there's a particle theory is that these outbreaks are unrelated, little sporadic outbreaks that are discrete independent events. That's one theory. The other one is it's a wave, that Ebola is expanding its reach through reservoir species. And there's a book called Spillover by David Quayman that I recommend. He's a reporter for National Geographic that really talks about this in great detail. But really, this is actually the same thing if you think about it, because these sporadic outbreaks you see in humans may be the result of the reservoir species spreading. And there's a lot of interesting papers that you can look about. If you Google the word wave-like spread of Ebola, you'll find some interesting papers. And I think it's really a fascinating, cutting-edge type of research, thinking about how Ebola is behaving in the reservoir species and when humans are getting infected. But this is the best that we know right here from this slide, where you see bats here. We see bats moving to gorillas, chimpanzees, for example, and also duikiers, which are like African antelopes. and then direct contact with humans that spill into humans, and then you have an outbreak when these individuals present at village healthcare centers, maybe get diagnosed with typhoid fever, get diagnosed with malaria, people don't wear the proper personal protective equipment, and then it spreads. And that's why healthcare workers in these village healthcare settings are disproportionately affected, is because they take care of these patients without knowing that they have Ebola, and then they also may have shortages of personal protective equipment. So that's what we thought: bats, chimpanzees, antelopes. Then this paper came out in Science a couple years ago. Discovery of swine as a host for the restin Ebola virus. So in the Philippines, where the first restin outbreaks were recognized in chimpanzees, they had these pigs that were sick with a respiratory virus, and then they also cultured a second virus, and they surprisingly found restin Ebola there. And they had never seen Ebola restin in pigs. That caused a lot of consternation because some of the handlers actually were turning positive, or testing positive for antibodies. So are pigs playing a role? And what does that mean for the virus? And I'll talk a little bit about that. So this was the paper in 2008. We had 6 human seroconversions, like the 4 we had during rest in Virginia. Pigs had respiratory disease. They were co-infected, but didn't really have symptoms. But the thing that was scary about this is that the Ebola restin in pigs had respiratory symptoms. In humans, you don't get respiratory symptoms. When you have respiratory symptoms with an infection, that gives you a whole new avenue for spread, because you sneeze, you cough, you have mucus. All of those types of things make it much more easier to spread if it is actually having a respiratory component to the infection. So that got people to do some experiments, and they actually replicated this whole thing with Zaire Ebola virus in pigs, and also got it to spread from pigs to pigs without direct contact. So that's kind of important to remember. Ebola in its natural environment does not spread that efficiently through respiratory droplets. It's usually blood and body fluids. It's not airborne like tuberculosis. But in these laboratory settings with pigs, there was some suggestion that you could do it in this species. And that caused people to write some papers. Ebola virus is a foodborne pathogen, cause for consideration, not panic. What I think was interesting from this is that, this whole transmission to humans via bushmeat, and we've seen some, I have a picture of the Newsweek article that really talked about that recently, got a lot of controversy, but bushmeat is a means where humans come into contact with viruses, and it's very important to study the blood of bushmeat hunters and those who consume raw bushmeat, because that's where you're seeing these viruses jump from. And if we were studying bushmeat hunters in the early 20th century, we would have seen HIV making its first forays into humans in Cameroon before it became able to spread efficiently from human to humans. It's important to remember there's no risk to eating cooked food. I don't want to spark a panic here like they did during swine flu. But one of the important things that they said here is the susceptibility of pigs to Ebola virus infection can also be seen as an opportunity for bioterrorists wishing to disrupt the food supply and infect humans or both. It doesn't take a lot to disrupt the food supply. You just have to hint at it, like Oprah Winfrey did with mad cow disease. It's a very sensitive group, and people stop eating swine during swine flu. People go bankrupt because of that. So this is a way of causing economic warfare. And I study bioterrorism at the Center for Health Security, so that's something that I'm very keen to, how Ebola might be used in a biological weapon. These are some pictures of some of the bats that are putative hosts for these Phytovirus. I think that's I've never seen a bat like that, but that's quite scary. So transmission to humans. I talked about bushmeat. This was a cover for the Newsweek magazine. I was actually quoted in talking about bushmeat a couple weeks ago, and it got a lot of controversy, but people do eat bushmeat, and there is a bushmeat trade in New York City where people have when I say bushmeat, I mean exotic animals that are eating them, that maybe it's beef jerky or delicacies. And anytime you really have close contact with these animals and you're butchering them without proper protective equipment, you get little cuts and things can get in it, and that's what the threat is for bushmeat. But how do humans get it from each other? It's close contact with blood and bodily fluids of infected, symptomatic people. Not people who don't have symptoms, like the people that are being quarantined in Mecklenburg County, North Carolina, without symptoms. They don't pose any risk because you can't get Ebola unless you're actually around somebody that has symptoms. Influenza, a day before you're sick, you might be able to spread it to individuals. But with Ebola, you have to have symptoms, and it's only blood and body fluids. So blood, feces, urine, vomit. Also, burial practices. If you read some books, you talk about what they do to prepare the bodies in certain West African countries. Some of them involve evacuating the bowels by hand. Those types of exposures, without wearing personal protective equipment, put you at high risk. Things that aren't really even thinkable in the United States. And what ends up happening when these outbreaks occur is that you institute very simple public health measures. Find the contacts, find the cases, isolate them, watch the contacts for 21 days to make sure they don't get fevers. Get the burial practices under control. Give people gowns, gloves, eye protection, and then these outbreaks stop. That's what's happened. It's failed here, and I'll talk about a little bit why it's failed here towards the end of the talk. important just to emphasize not contagious prior to symptoms. And I think the symptom severity prevents spread. This isn't like influenza where you can walk around and go to work. It's not like that. You're sick. It's very hard to spread it when you're bedridden and dying. So that's another kind of a chink in the armor of Ebola, that the very power that it has to kill someone is also limits it in how it can spread. Virus can be seen in the semen for 3 months, and so people are advised to practice safe sex after they've had an Ebola infection. And that became an issue with Dr. Brantley after he was discharged, but they had made a big point of saying they counseled him on this. So this is a question I get asked a lot by the press. Is Ebola airborne? Reemphasizing. Transmitted through blood and body fluids. Some suggestion that restin may have been transmitted between monkeys without direct contact, but monkeys don't have to directly contact. They tend to throw things at each other. They tend to throw their feces at each other. It's hard to say exactly what happened, but we did see some suggestion with pigs as well, because in pigs it is a respiratory tract infection. In human, it is not a respiratory tract infection. So naturally, Ebola spreads through humans through blood and body fluids, not airborne route. So that's why we're emphasizing droplet. That's why, we're saying when the CDC talks about how you take care of these patients, any hospital can do this, and you need really droplet precautions. There are some hospitals that are using airborne precautions out of an abundance of caution. And when anybody uses the word abundance of caution, you should get your ears should be up, because they can say a lot that it means that they're doing something that isn't quite science-based, but it is understandable because people are scared of this virus that we may be using, that people may be using airborne precautions. And it's unfortunate that those individuals that were treated in Emory and the new individual in Nebraska were treated in such a high-level containment facility, because it gave the wrong impression to the public about how Ebola can be treated. And this is a biocontainment unit that happened in Hamburg. So I'll talk a little bit more about this when I talk about vaccine, but somebody got needle stuck in Hamburg, and that's the kind of containment unit they use. But a lot of people do this, but you don't necessarily need to do this. It's just what has always been done with Ebola, because people have been afraid of it. But we do take care of this in normal settings, and even Dr. Veldkamp in the crowd took care of a Lassa fever case, probably in less contained areas than what you have to do for Ebola, and that spreads in a very similar manner. So we're going to move on to the clinical features. So the incubation period is 2, it's a typo, it should be 21 days, 2 to 21 days, but most get illness by 10 days. Fevers, myalgia, headache are often the early symptoms. People also get nausea, vomiting, diarrhea, abdominal pain, pharyngitis. They can get delirium, somnolence, coma. 50% bleeding manifestation. So not everybody is bleeding from their eyeballs, bleeding from their injection sites. That's important to remember. And it may be that those that are having these symptoms of bleeding are the ones that are more contagious and driving the epidemic, and that's an active area of research, is who inside the Ebola outbreak are the ones who are the super spreaders. Like in SARS, we had super spreaders, people who disproportionately infected other people. Typhoid Marys. It'd be interesting to figure out who the Typhoid Marys are of Ebola. People get a rash. They can get some lab abnormalities. Electrolytes may be off. Blood clotting will be off. LFTs. They can get leukopenia. And usually people die in the second week from multi-organ failure. This is a slide just showing you days to death after onset of illness, and as you see, it clusters around this early second week of illness, but we do see some outliers as well. So how does Ebola cause its disease? So it enters mucosal surfaces, breaks, abrasions in the skin, or through parenteral introduction, so needle sticks. It has many target cells, monocytes, macrophages, dendritic cells, all these cells of the immune system. And this triggers a huge immune cascade, which is responsible for the symptoms of the disease. It's kind of like septic shock. A lot of this has to do with your immune host response to the virus, rather than the virus itself. It does cause apoptosis, or cell death of lymphocytes. It can also do the same in liver and adrenocortical cells. And this is a picture of a liver showing you necrosis starting to occur in a liver of those infected with Ebola. This is a slide kind of showing you some of the schematics of how this virus causes infection. It's just important to remember it has multiple different target cells, and does cause lymphocyte apoptosis, can cause liver damage, and also just really causes a huge immune cascade, which is responsible for the symptoms. So viremia correlates with the clinical picture. Just like in HIV, you think of the viral load. When people have high viral loads of HIV, they tend to be sicker. Same with Ebola. And when you start to get antibodies, the viral load comes down. Protection, though, it's not considered to be, it's somewhat antibody-based, but it's not completely clear, because we've got some conflicting data on convalescent sera. Some people say that it's works, some people say it doesn't work. It is going to be tried again. Kent Brantley did get convalescent sera, so it's something important to think about. What is the protection from Ebola? Is it antibody-based completely, or is it cell-mediated immunity? It's got a lot of virulence factors we talked about, the glycoprotein, the fact that it causes extensive lymphoid damage. It also totally screws up your interferon signaling, so your body's immune system is going haywire. It also activates tissue factor, which causes clotting and disseminated intravascular coagulation that you might see, which can lead to the bleeding manifestations that are so dramatic with Ebola. So we're trying to figure out who lives and who dies from Ebola. And this paper just came out right a couple months ago in Emerging Infectious Diseases. And we found that actually children and adults are different. They have disparate biomarkers. So children have different biomarkers that may be elevated or low, and they survive versus adults. And this is going to be an important thing to figure out is how do you know who's going to live from Ebola? And I think they're going to see much more studies on what biomarkers you can use to gauge someone's likelihood of surviving Ebola. So now moving to diagnosis. How do you diagnose Ebola? Usually it's going to be clinical. It's going to require an astute clinician. If a case shows up here in Presby's emergency department, you have to think, did this patient go to somewhere in West Africa? Were they in contact with someone from West Africa? So I can't emphasize that enough, that you think about Ebola when you ask people their travel history. You should be asking travel history for everybody, because it's not just Ebola. We've got MERS going on in the Middle East. We've got chikungunya going on. Every day there's something that's somewhere. And it's important as an infectious disease physician that someone asks the travel history, because people travel all over the place. So other lab things you might look at: thrombocytopenia, leukopenia, elevated LFTs, coagulation abnormalities, and then the clinical triad: rash, abdominal pain, diarrhea. It's important to know also that here in our protocol, we'll be doing bedside lab testing. We will be using our iStat cartridges so that we don't send things down to the lab, and that's sort of what other hospitals are doing to avoid contamination of instrumentation with blood. So that will be, I'll talk a little bit towards the end about what we're doing here at UPMC, but it's important to remember that we will be able to do blood tests, but at the point of care. Microbiological confirmation is mostly going to be done by PCR, and that PCR is going to be done by the CDC. The State Health Department can't do it. It's only the CDC that really has the primers. You can do viral culture, serology, staining of tissue, but that's not going to be relevant in this setting, but it is relevant in other settings in Africa, for example. This is a slide just kind of showing you the different diagnostic tests, and I just want to show you the PCR is the one that it does require specialized equipment, but it could be used in the field laboratory. It's rapid, it's sensitive, it distinguishes between stains. It's the diagnosis of choice to confirm Ebola. And that's what happens when Mount Sinai has a suspect case. They send the PCR to the CDC. There has been an emergency use diagnostic that's been it's been waived for FDA approval, has gotten emergency use authorization. It's a Department of Defense kit that allows you to test for Ebola Zaire for real-time diagnosis. It's not probably the one that's being used at the CDC, but it may be what's being used in the field now that the Department of Defense has deployed to help with logistics in this West African outbreak. So we're going to move on to treatment now. So what you hear is supportive care, and supportive care may sound generic, but it actually makes a big difference. There's a lot of suggestions that IV fluid, aggressive IV fluid therapy lowers mortality, and that's something that we know just from ordinary sepsis cases. And one of the biggest cornerstones of the Surviving Sepsis campaign and early goal-directed therapy for sepsis that we all should be doing is to give people IV fluids, and that seems to be a big factor in whether someone lives or dies. It may not always be available. IV fluids might be scarce resources in parts of Africa, not so scarce here. That's something that you need to remember, that may be why we're seeing this 50% mortality rate with this, because maybe people are getting more access to IV fluids. Fever control, antiemetics. There is some suggestion that convalescent plasma works, taking plasma from those who have survived. Dr. Brantley did get convalescent plasma from a 14-year-old boy. There are some novel products that I'm going to talk about: ZMapp, the Takerna product, and the Biochris product that all have seen a lot of press and some use in this outbreak. So moving to convalescent plasma, this is a paper from 1999 from JID, where they did use it there, and people did, lower than they had only one transfused patient died, lower than the normal overall case fatality rate. But it's unclear. You have to really make a decision whether or not this is the convalescent plasma doing it, or are people getting better care? Because we're not doing randomized controlled trials. There's lots of confounding variables. Are people that are getting convalescent plasma being treated by more aggressive doctors? Is there something different about their treatment than other people's? All these confounding variables that need to really be sorted out before you can say for sure that convalescent plasma works. But this is a good opportunity to try it. The WHO is encouraging blood banking to occur to be able to do this in this outbreak setting. So we're going to move to ZMapp. This is the one that's gotten the most press, the most interest. I actually worked on a Department of Defense project with the guy that invented ZMapp, who was a graduate student with one of my colleagues, and decided to work on Ebola because nobody else was. And then he actually came to our meeting and started talking to the Department of Defense people, and they got very interested in this product. So we kind of have a lot of interest in how well ZMapp does. So what this is a cocktail of 3 monoclonal antibodies directed against Ebola's glycoprotein gene. And it has really compelling, great data in non-human primates. They are exposed to Ebola and they live, even after illness in the late advanced stages. So this is as close as we can get to human data. It's been administered to 7 individuals so far. And we've had some people live, there's been people that have died, for example, the priest in Spain. So it's important to understand what the window is that you can give this drug in, because people may be too far gone, just like in septic shock, to actually benefit from any treatment. And there are a lot of confounding variables. When Dr. Brantley received this, there were people on TV, Sanjay Gupta, namely on CNN, saying this was miraculous. In 20 minutes, he was better. And then you had Tony Fauci from the NIH saying that's not how this works. It couldn't have been that. So it's important to remember that there's a lot of confounding variables. This guy got plasma therapy. This guy was already he was ambulatory by the time we saw him in the United States. He was never intubated. He may have gotten IV fluids. So it's hard to say what role ZMapp made, but I don't want to discount ZMapp. I think this will change the face of outbreaks in the future, but it needs to be studied in a way that actually proves that it's effective and safe. Some other drugs to talk about are the Tecmura product. This is a cool drug. It's an RNA inhibitor. So this is a piece of RNA that goes and silences the genes of Ebola. We only have one other infectious disease drug that's an RNA inhibitor, that's Phytovirus for cytomegalovirus. So this is a great drug, and it's a great step forward to use these targeted therapies against pathogens. And I think we'll see much more of RNA inhibitors used for both bacterial and viral stuff, and it's great that this is seeing some light of day. There were phase 1 trials that had been started prior to this outbreak. They were stopped by the FDA because people were getting flu-like symptoms. They've now been resumed because the cost-benefit has shifted now because of this outbreak. BioCryst also has a product, which is an RNA polymerase inhibitor. People are probably familiar with RNA polymerase inhibitors, and that's also in trials. But there is a whole host of drugs that are in the pipeline, and just a list to show you that there is a robust research agenda. There's an article here about high-dose nanos binding lectins. So lots of people have been working on Ebola for many years in the biodefense community, trying to get countermeasures because, and I'll talk a little bit about its role in bioterrorism towards the end, but this is a pipeline that's been developed basically since September 11. So, how to advance now. So vaccines. So you're hearing about this vaccine that's been donated to Canada, from Canada to that area, and this is the one that's probably the most promising. It's a recombinant vesicular stomatitis virus. This vaccine takes this virus and puts in the glycoprotein gene. Again, another mention of that gene, how important it is for Ebola. And it's been used actually in one human, which I'll talk about in a second. And this is probably one of the most advanced ones. The NIH and GlaxoSmithKline are now doing a trial with Chimpanzee adenovirus vector vaccine. You might have heard of adenovirus vector vaccines for HIV, for example, which failed. But this is a Chimpanzee one, which is supposed to have some advantages that the human version didn't. Bavarian Nordic is working with the smallpox vaccine and has engineered to have the glycoprotein gene in as well. And then there's a Marburg Ebola combination vaccine that Cristal is working on. So there is several vaccines that we are seeing development of. Talking about this vaccine, the Canadian vaccine, this was the accidental lab stick that happened in Germany in 2009. And they had this big conference call, what are they going to do, what are they going to do? And they actually administered this vaccine. This is that person's case. And you can see here that she had RT-PCR positive for the glycoprotein. It's hard to say whether that's the vaccine or the virus. And the VZV RT-PCR was positive as well. And they followed her, and you can see she gets it, she gets a needle stick, they give her the vaccine, they put her in the biocontainment unit, they take her out. Then her D-dimer starts rising. That's a measure of clotting. They put her back in. She gets a little bit of a low-grade fever spike. Every time she gets a fever spike, they put her in the biocontainment unit. And she did well. She didn't get infected. It's unclear whether that needle stick was significant or not, but in that situation, they had to give her something, and they tried the vaccine. So this was the first human to receive that vaccine. This was published in a big supplement in JID a couple of years ago. So now moving to biowarfare issues, and this is where I really am very interested in Ebola because that's a lot of the work that we did. The think tank I work at was founded in response to the threat of bioterrorism when we were intimately involved in the anthrax attacks. So what we knew after we had these series of Soviet defections is that the Soviet bioweapons program produced large quantities of Ebola in Marburg. Some of them say it was for vaccine research, but it was clear that people were interested in this as a bioweapon as well. And they realized that a low infectious dose, maybe even one virion, can cause infection. And that's why it becomes attractive as a biological weapon. There's also some indication that the Iraqi weapons group was also looking at Ebola and Marburg as bioterrorist weapons. The Japanese cult Shinrikyo used to send people to Ebola outbreaks to get samples of blood. So this is something that really people thought of as a bioterrorist weapon. And because of that, after September 11th, the Bush administration thought, we have no countermeasures against Ebola. This is something that we know that's been tried before. We've just been hit with an anthrax attack in which 5 people were killed. They started a whole biodefense enterprise of funding to start to get countermeasures against this virus. And what we're seeing now with ZMapp, with all these products, these vaccines, is really the fruits of that effort. It started in 2001 to prepare the country for bioterrorism. And that's an important thing about bioterrorism. When you prepare for bioterrorism, you're preparing for all hazards. These things that were thought to be using in case we were hit with an Iraqi weapon using Ebola are now being used in an outbreak setting in West Africa. So this biodefense funding is paying dividends in another area. Another couple slides I wanted to show you, titles is, there was some backlash that when the United States started looking at biodefense for Ebola, there was a backlash. It's kind of how different an outbreak, how different it is now. That people are saying that, this is a huge public health threat, it's a neglected tropical disease, and people need to get more robust funding. But back then, they were saying this is really a relative minor public health threat for Africa, and this is just really being pushed by the biodefense agenda. So now everything has changed. Now people saying, we want these drugs, we want all this experimental drugs. So it's kind of, you can't have it both ways. But it's an interesting change in the dynamic of the argument that's been going on. So now the last part of the talk, I'm going to turn to what happened in West Africa. So as I alluded to before, this was the first Ebola outbreak that we've had in the region. There was just a one-off case in 1994 in the Côte d'Ivoire. But it's probably been there at low levels for a while, and there's been some, I'll talk a little bit about one of the papers that came out showing some antibody prevalence that have been found there. They do have another hemorrhagic fever there called Lassa fever in West Africa that is very common, and sometimes they empirically treat people for that. And some of those cases may actually be Ebola. There's a very huge lack of infrastructure in this area. There's said to be only 50 doctors in all of Liberia. There's massive personal protective equipment shortages and absences. People are intubating patients. We heard a story in Baltimore at my place of work there that from the president of the Liberian Medical Society, that people are intubating patients because they have to quickly intubate when they don't have the personal protective equipment to do it. So they're left with the choice: do I let this patient crash, or do I put a breathing tube in them without wearing a mask? And that person got infected and died. That was the famous Dr. Khan, I believe his name was, that died in Liberia. So people have a real dilemma whether they take care of the patient without the personal protective equipment or they let that patient die. And people are reusing thermometers there. That's something that's really unthinkable here. People don't reuse thermometers in their own house here. This is crazy. This is kind of something that's really unfathomable by the U.S. population about what happens in these healthcare settings, because it's not, and that's why the fear of Ebola spreading in the United States is very overblown. It cannot spread in this environment because it's totally inhospitable to the spread of it. There's a huge distrust of authority. These countries, some of them emerged from civil wars, they have dictators, they have such a mistrust in their government that they aren't listening to simple public health communication messages. People are running from Red Cross trucks. There was a great New York Times article that talked about that. So this is something that's really unique. It doesn't happen in these outbreaks that happen in DRC. The border is the epicenter, and that also causes problems because people are moving from country to country. They're hiding bodies. Bodies are moving from country to country, from one country to the other. It just makes it very hard to do the simple public health measures that you need to do, the contact tracing we do just for a tuberculosis case, for example. There are some authoritarian government measures that often backfire. I wrote an op-ed for the Washington Post about this. When they quarantined the West Point area of Liberia, they were shooting people. That causes such mistrust in the government. It becomes a dead zone where nobody can even get food or water, and it just makes it an area where the virus can find more places to spread, when dogs are eating bodies, when people are looting Ebola hospitals in these quarantine zones where there's no law and order. So quarantining healthy people is never going to be the answer. So one of the interesting things, this is from the New England Journal paper, that patient 0 was a 2-year-old patient, which is really, usually it's a hunter or somebody that's been out in the forest and has butchered a chimpanzee. That's the first case. This is a 2-year-old. So there may have been an unrecognized case prior to this 2-year-old, or maybe this 2-year-old came in contact with a dead bat. Maybe they ate a piece of fruit that was contaminated by a bat. That still remains to be seen. And the virus forms a whole separate branch. It's in the Zaire branch, as you see here, but it's its own. See, this is Sudan, there's Reston, there's Bundavugia, there's Thai Forest, and this is the Zaire branch, and it's a whole new lineage here. And it's likely that this has been circulating for a while, and it's gotten some of its own characteristics compared to the other parts, the DRC strains of Zaire, the Gabon strains as well. Most people think it's probably been in West Africa for a decade or less. Maybe fruit bats brought it there from other places. There are mutations accruing. This paper just came out in Science. This virus is starting to show mutations there. nothing really showing that it's increasing its virulence. If anything, it's showing itself to be less virulent than prior outbreaks. So this is what I was talking about, undiagnosed acute viral febrile illnesses, which mostly were Lassa fever in Sierra Leone, in the Lassa fever clinic. 9% of those individuals were seropositive between 2006 and 2008 for Ebola. So Ebola has been there, just kind of lurking beneath, unrecognized, until it got the opportunity to spread like this. So there are some differences between the countries, and what we're seeing in some places is exponential growth curves, but that's not the same in every place. This is a slide talking about the R0. So if you've ever watched the movie Contagion, the Kate Winslet character goes on the board and talks about how what an R0 is. What that is how many people get infected by one individual. So measles, maybe 15 people will get infected by one case. Flu, maybe 2. So it's important when the virus's R0 drops below 1, it can't transmit because you're not transmitting it to everybody. So anthrax, which is not contagious, its R0 is 0. So what we're seeing in certain countries is the R0 falling again, which seems to have much more control, at least anecdotally. And the R0 staying about the same in Liberia, 1.5, and starting to fall in Sierra Leone as well. This is very hard to actually gauge. There's a lot of assumptions that go into this, so these may not be valid. The point I'm trying to make is that the R0 is going to really vary depending upon what the infection is doing. Are these people vomiting? Are they having diarrhea? Are they being isolated? What are the patient characteristics? Is this a typhoid Mary that we have, or is this an ordinary case? And it's important to know that once you can tamp down the R0, you can stop an outbreak. So that's what measures do. So example, in influenza, people talk about closing schools because you decrease contacts, you decrease potential for spread. That lowers the R0. A slide about experimental therapy, since this is a health equity talk. So there's really some issues here. These drugs have really never been used in humans before. We don't have any long-term safety data. And it's very hard to give informed consent in this setting. So these are things that people need to think about. It's not just a reflex to give people these drugs, because what would happen if a side effect happened? And then the United States would be accused of experimenting on African people with an experimental drug that had never been used in Americans before. So I don't think there's any way to win this on either side. Either the Americans are getting favored treatment, or if we gave it to everybody, then you're experimenting on African people. Just think of the movie The Constant Gardener, with Rachel Weisz, where they give this experimental TB drug and people die there and there's this big cover-up. So I don't really think you can win with this. So it's really important to know, what are you going to do when a safety signal emerges? Just think about Vioxx and drugs like that. When you give it to large amounts of populations, you might see side effects that you don't see in non-human primates or in small groups. And this is a very scarce resource. We're actually out of ZMapp right now. The guy in the OBGYN that's in Nebraska is not receiving ZMapp because we don't have any. This was a drug that wasn't even in phase 1. So a couple other questions. So lessons for a couple other last slides. Lessons for the U.S. Like I said, Ebola thrives in environments where blood and bodily fluid exposure is rampant. It is not a very contagious disease. It is deadly, it is not contagious, and there's been a confounding of the word contagious with infectious. Not all infectious diseases are contagious. Some are more contagious than others. Measles is probably the most contagious. Anthrax is not contagious. You have to think about it as a continuum. We've had multiple importations of Lassa fever in the United States, 8 of them. No secondary spread. Nobody knew these were Lassa fever cases until they were diagnosed retrospectively. We're pretty good at managing these types of exposures in the United States. If you can import Lassa fever unbeknownst, without any public awareness, and not have any secondary spread, that means what we do well. I know we all complain about you guys not wearing your gowns or not washing your hands and not putting TB patients in isolations, but I think we did okay here. We also had the importation of Marburg, no problem as well. We've had 2 importations of MERS in the United States, which everybody's forgotten about, but MERS hasn't forgotten about us. This is much more contagious, and we had no secondary cases as well. So I think that we will not find this virus, finding the U.S. to be a hospitable place. I don't think England or any of the westernized countries will not see Ebola cases spread, other than maybe an imported case. But that doesn't mean you shouldn't be vigilant. I think we have to be prepared here to have an infected traveler. I know for a fact that we have missionaries from Pittsburgh that are in that area. They may come back eventually. So we have to be able to ask people their travel history, and I just want to emphasize that again. There's always something going on somewhere in the world. Ask travel history in everybody. Rule out malaria. I just don't want people to think Ebola, 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 and then someone dies of falciparum malaria. Malaria will kill you, and you need to think about malaria in any infected traveler with a fever. Call infectious disease. If you get scared, if you think something is odd, call infectious disease. There's a pager 24/7. Somebody is there. Call infection control. We've got representatives from infection control here as well. We are working on a protocol. We're going to be revising it. It's available on the InfoNet. Don't Google. Go to the info. People tend to Google without looking to see what we've already had. We've got everything specific for UPMC because we have certain health department numbers to call that are not going to be the same as what you find on Google when you see some other hospital's protocol. It's better to use our own. We have the right chain of command. We need everybody to be involved and on the same page, because if we do get an infected traveler, we want this to run as smoothly as possible. It's going to be hectic, and the more just uncertainty and nervousness that occurs because people are doing their own thing, it's going to make it much harder to control, and we may end up with secondary consequences that we don't really want to have. That's why we make our own protocol. And Kathy Chris from Emergency Management is sitting right there in the third row, who's handling all of that for us. And I just want to emphasize, we don't need to do the full containment like Emory and Nebraska did, but that's something that everybody is doing. This is my last slide. This is a picture of them drying their gloves out in Africa, and also can you raise hands for questions if you have any questions. I urge you can email me. You can. I have a blog called trackinzebra.com that I encourage people to look at. I talk about these types of issues, and you can also follow me on Twitter where I talk about Ebola and other things as well. So thank you for your attention, and I'm happy to take any and all questions you may have.